All right, so thanks everybody for coming out tonight. Really excited to introduce Chris Ng, one of the co-founders of Protein Cure. So it's a really exciting startup in the protein design space. I think there's not a lot of companies using computational protein design out there nowadays, and yeah, I think Protein Cure is doing some really exciting stuff, pushing the boundaries. So yeah, Chris, please take it away.
Thanks, Chris. Thanks for having me. Really appreciate the opportunity to share kind of this fun style talk, which is sort of an overview of, a lot of different work done in different areas of, bringing protein therapeutics to the clinic. So this is in the style of kind of a self-help seminar. You put yourself in the shoes of a protein therapeutic, and I'm going to guide you through some lessons, mostly structure-based ones, about how you can succeed, on your ambitions to become a great therapeutic. So that's kind of the twist. So the little forward at the start, I'll just say that, it's a startup company we're working in. I'll talk a little bit about it at the very end. We work on proteins and stuff like that, but the opinions and generally of this talk are really just of my own, just kind of an ingestion, a lot of papers, and especially that most of this work is published and the images are owned by the respective copyright owners and such. And I followed the citations to get more detail on some of the things I mentioned. So we can start to dig right into this, problem of being a protein therapeutic and aspiring to be great. And where we look to see our heroes in this space, well, mostly as stock photography on biotechnology websites. We see, our heroes in families growing old together, in, families working to teach, let their children learn how to ride a bike. But how do you get there yourself? If you're a protein therapeutic and you're just starting out, sometimes it can take quite a bit of thinking, introspection about how you can get to that final place, because sometimes they're few and far between, like heroes to aspire to. And sometimes you can look high and, well, low and high in order to try and find out what you should do and what path you should follow. And so that's why I kind of formulated this talk to give protein therapeutics some strategies to move forward in the world and advance on their aspirations, really to reach the clinic. So the overall objectives of the talk are to give you these strategies. First of all, some motivation for each one of the 7 kind of habits, as I'm calling them, that you should have. And I'm going to kind of shoehorn a lot of examples into each of these habits and give you sort of a little bit more an understanding about how this kind of like this abstract and, amorphous idea or like this habit can translate really to examples of success. So let's just kick it off with the 7. I mean, I've got 7 and we'll just talk about each one in detail. The first one, is one that's pretty general as well, just to be proactive. That's just taking responsibility for your reaction response to your experiences as a protein, recognizing what you can influence and what you should be concerned about. And so let me translate that a little bit into terms you might be a little bit more familiar with. So you can interpret that as being a successful therapeutic by being optimized for your administration route, oral, inhaled, transdermal. To be a great therapeutic, you have to be mindful of that and have great intrinsic stability properties. You have to be concerned about your bioavailability, because in the end, it results in convenient dosing for these patients that are taking this drug. So while there are things you can do to yourself, to your structure, to your design that can assist in those things, you as a successful protein therapeutic also have to recognize that there are things outside of yourself, opportunities for fusion to other proteins that can improve your delivery to tissues, cells, or even subcellular localization. So let's just dig into some examples of that. And I should say that there's probably some great examples that I'm completely missing, and I'd just love to hear about them too at the end of the talk or throughout if you wanted to make a comment. But starting left to right, you can see some examples of getting out there. That would be optimizing your surface for enhanced solubility. There's an example of monoclonal antibody where this patch of hydrophobic residues was removed through site-directed mutagenesis. Often structure-based methods are used for this purpose. And even just going beyond natural amino acids, there's lots of strategies to overcome serum proteases and improve against protein degradation, as well as detection by the immune system. By deimmunization through removal of T cell epitopes or otherwise. Here's an example of this VEGF protein that was developed. And it was highly successful when it was inverted in a mirror image from L to D chirality. So that's just a successful example of achieving that. And another one you may be familiar with on the protein design side would be just enhancing stabilization, repacking your protein core, adding additional constraints, cyclizing. These are some classic strategies that were put in, demonstrated well in this paper that Chris actually contributed to at the Baker Lab. Many of you have seen. And beyond just being more soluble in general, having better properties to move through the body, there are places where some proteins can't go. This is a problem as you become a bigger and bigger protein, but there's a boundary in which small molecules transition to these sort of cyclic and macrocyclic peptides, where there is an interesting balance between conformations of the peptide, which may be more suitable for cell permeation, and some that won't. And so that's even another topic we discussed at the meetup. Parisa came and gave some nice examples of her research in that field. And this is an example. This picture just shows cyclosporin, which is, well, one of the most well-studied examples of this phenomenon. It's not every day you can design yourself to have these properties, but we're getting there. We're getting there to the ability to design peptides with chameleonic properties. They can have their solution state and lipid state conformations tuned through potentially something like multi-state design. So these are some intrinsic things you can do to, take initiative. But there are also these strategies in order to extend your half-life in the body, such as fusing yourself to an IgG Fc domain or human serum albumin to, take your half-life of your protein from like maybe minutes all the way up to many days or weeks. We've seen like successful examples of that in the type 2 diabetes space many times. And I think this is the first of two examples I show of optimizations made to this recombinant protein, the the what is it, F7. I guess it's a blood clotting factor. It's a highly effective therapeutic for treating patients who have issues with blood clotting. So that's one strategy you can go with. And there's many examples like oral available insulin and oral available GLP-1, again, in the type 2 diabetes space, where pegylation, that is just attaching essentially a fatty acid to your protein structure. Here's this like oligomerized insulin hexamer or a trimer of dimers, I believe. And it is also just a very effective strategy for this purpose. Now, getting inside the cells is one thing, but getting to difficult parts of the body, such as past the blood-brain barrier, is a major challenge for protein therapeutics. There are numerous strategies, one of which, like receptor-mediated transport, which is being used effectively by the pharma company Denali to treat neurodegenerative diseases, is one great strategy. But there are even some simple things like adding peptide tags, like this is 20 amino acid blood-brain barrier tag, which again is receptor mediated. It binds to a target in that space and is allowed to be uptake. So that's an example of something we worked on in Protein Cure, but it's of course done numerous times outside of Protein Cure, where the problem is sometimes where you need to attach this tag and what kind of linker you need to use in order to make sure it is flexible, but also not too flexible, such that it's just cleaved. Lastly, there's just numerous examples of very specific cell targeting strategies, such as this one used by Ipsen, which is a fusion of a protein, like this is the payload or some cargo that you're delivering to, in this case, very specific neurons. So this uses this Botox delivery strategy, and there are a few other platforms like this that are like the foundation of many pharma companies. So that's just a lot of talking about ways in which you as a protein therapeutic can get throughout the body better. But there are many other strategies you need to do in order to be, not just in the presence of your drug target, but to bind to your target effectively. So a good strategy for that is this habit, beginning with the end in mind. And this is not something that's probably familiar to most people who work in structure-based design. Envisioning what you want in the future so you can work towards it. It's not always possible. You don't always have a structure-enabled project, but when you do in drug discovery, there's lots of great strategies you can take, especially because we're protein designers. We can think about that. So as a protein therapeutic, the knowledge of the target has really a fundamental benefit. It can help you, hone in on a specific functional epitope of interest. Functional binding sites are, of course, key, but they're not always, and it's not always the end of the world if you bind to another position, if your job is really for localizing to a specific protein on maybe a cell surface. But co-crystal structures are naturally best. They allow you to work on kind of a, a motif grafting approach, taking those critical interactions in an interface and grafting them onto other scaffolds. There's also sort of another bottoms-up strategy where you can sort of assemble fragments. And this allows, again, you the benefit of being able to mimic known protein-protein interactions. This is something that protein therapeutics have that small molecules, kind of wish they had, which is just the ability to have a starting place that is something that's, where it's possible to measure, cellular pathways and responses to these type of perturbations and modifications with protein-protein interactions. So, with the ability to perform structure-based design, naturally the first thing you might want to do is optimize for novel hits or just identify new hits and optimize those hits. Potency and selectivity would be kind of the first things you might want to get. And well, there's probably a gazillion examples of that. Some of them you may have seen, the work that started the biotechnology company Neoleukin, the Silva et al., where they designed this protein binder, strategically eliminating a binding site to make it a better therapeutic. And we also have examples, from the peptide space where they're natural helical peptide binders, which are enhanced by structure-based design, allowed us to install at strategic positions staples like these hydrocarbon linkers, which allow the secondary structure to be enforced, but also allow the staple itself to form sometimes contacts that are beneficial for binding. So this was one in which was a peptide for the MDMX and MDM2 targets. It's like a P53 analog that was formed a lead candidate for aileron, which is still undergoing. It's been going through a number of trials. Here's another example of the use of structure-based design, which I think is kind of like, great, is that you have with the computational method an opportunity, if you're essentially looking to bind to a target, you can bind to many places in some cases, if you're not specifically trying to target a functional position. And that allows you to have, lots of options for lead candidates of, different scaffolds and at different positions, which even offers you the opportunity to link them in some way later down the road if you should so choose. That's another topic we'll discuss. Lastly, I just was trying to find an example of structure-based design where selectivity was like a key driving force, and that's kind of, in many cases, that is true. Here's one from, I believe, Poly4 Pharmaceuticals. That's it. And it is a beta hairpin cyclic peptide based on their platform for developing antibacterial peptides. And so as you can see, it started with this natural peptide PG1 when it was successively mutated to improve its selective binding properties to both outer membrane protein beta barrel, as well as even I guess sugars on the surface of the sugars as part of the cell membrane. Okay, so those are examples of structure-based design. I think we could advance too to this other topic, which is something you can do as a protein, is just prioritize. Because there's sort of a zillion things you need to optimize and do, but it's important that you need to recognize what is important to do first, plan, delegate the rest, eliminate things you don't need to do. Maybe that's kind of obvious, but it's pretty important to have a methodological approach about this, and many pharma companies do have that as part of their drug discovery pipeline. But it is kind of different sometimes for the specific protein target every time. Um, effective protein therapeutics have to have their priorities straight when it comes to go no goes. And if it's the first in class protein, like the first protein to bind this particular target, you may not be so concerned with certain things like the specific binding epitope. But if it's a well established target where the binding epitope is known, you may want to differentiate yourself, especially in the antibody space where there's maybe a lot more commonality between biologics. So for IP purposes, that's a good strategy that you might want to prioritize early on to make sure you're not developing and investing a lot of effort into designing a peptide or protein binder that is kind of like already been done. There's even situations where the scaffold or modality of choice might be specific or better suited for the target than another. We have the opportunity as a protein to adopt many different folds. I mean, small molecules have the opportunity to hop different scaffolds, but we also have the opportunity to hop different fold types from, helical bundles, which may be better suited for some computational methods, to, all sorts of things, all sorts of little mini protein folds. But in general, like you want to make sure that you are differentiating yourself as a therapeutic early on, especially from your competition in the pharmaceutical competitor landscape. And so that might mean successive optimization in a particular path or joint optimization of some of these properties. So, there's some interesting things. You might always want to be a better binder and a more selective binder, but there is a point in which you might not have to be any better on this property and you can actually work in some other area. So here's some examples of that. So there have been lots of, in well-established targets like this particular HER2 protein, there are highly successful antibodies, trastuzumab and pertuzumab. And an important differentiator for this protein therapeutic that's shown here was to be able to confirm that the epitope they were binding was fundamentally different than those. So that is, I guess a key prioritization for that study. As far as modalities go, especially for mini proteins, there's numerous ones. Some of them are only available through licensing agreements with particular pharma partners, but some of them are just kind of like classic protein folds that are not, patented in some particular way, and they might have certain size criteria or certain ways in which they display mutatable functional surfaces, whether those be loops or whether those be kind of on some type of functionalized helix. You can sort of tune that. Another example of just an example of that problem of not always having to be the best binder is that there are situations these days where if you are highly specific, you can compensate in other ways. And one of the classic ways to do that, which only recently was kind of demonstrated well by this paper lead in 2020, they took a regular old protein interaction, like this is a natural protein in the protein drug that's PD-1. This is like an interaction that's important for checkpoint inhibitors, and they basically made a non-canonical amino acid insertion that allowed covalent linkage, which gives you sort of infinite binding affinity. And this is like already a natural interaction, so it's pretty well tuned. There's, in this case, not really much likelihood that you're going to covalently bind to some other thing in the cell. So that's also an area we're quite interested in, proteins as covalent inhibitors. Something that small molecules have kind of a track record for with all their kinase inhibitors, but not something proteins are known for. Lastly, there's this problem, of course, of differentiating by early on by looking at your immunogenic risks and dehumanizing where possible. Again, the blood clotting factor here, an example of removal or a single point mutations that really prevented basically anti-drug antibody formation in, or probably not done in a, well, there's a number of assays that they can use to measure this, but this is kind of a work in this space for dehumanization can be critical early on. Okay. Now we're moving on. So the fourth habit, thinking win-win. So this is going a bit above just yourself as a protein therapeutic and thinking about the bigger picture. You want to search for mutually beneficial solutions or agreements in your relationships, value and respects those with competing interests. So in this case, you as a protein therapeutic might just want to get to your drug target, but there are competing interests at play here, mostly business interests as far as I see it. So, well, not necessarily just business interests exactly here, because great therapeutics kind of preempt developability problems downstream. So, for example, scaling up, and a lot of these problems in these processes can be a result of just not being able to be produced in high enough quantities, and it could be that trade-offs could have been made earlier on in order to kind of optimize a bit for those downstream processes. The sooner you can be testing these manufacturability things, the more likely you are to get through the whole drug discovery gauntlet. So that could mean, looking at that synthesis and expression, purification, things even like how you might formulate yourself at least in the biologic space is quite common, CMC. And this is just a limit. It's just however far you can do this is only really governed by what kind of in vitro and in silico models you have of these processes. For example, in biologics, there are certain properties that most proteins wouldn't think about till way later on, like viscosity, or in small molecules, its formulation, like how shelf stable are you? In peptides too, like there's lots of these issues, which don't seem initially like your responsibility as a protein therapeutic focused on your target, but something to be mindful of. Diversification early. So some examples of that are interesting. Often these examples you don't see them front and center, like this is a protein that made it to market because of so-and-so was mindful of developability. You often see it in kind of these retrospective studies, or in this case, two examples of just peptide-based studies that are kind of interesting. Improving peptide synthesis efficiency with machine learning is something that was done recently. Pentalute lab, I believe, at MIT did a nice work on this, integrating lots of data they have on the efficiency of coupling amino acids as you build the peptide sequence in solid phase synthesis. So that's an example of thinking about, being able to predict when your synthesis might be bad or things that are difficult. Certainly would be difficult to scale up if there was a problematic coupling or aggregation forming in peptides. In here, one protein cure worked on just balancing something that is seems like two separate worlds completely. The predicted potency of a therapeutic with the synthesis costs of producing it. Synthesis costs in this case, you can almost always you can get custom synthesis or find a vendor to supply you with an amino acid, a non-canonical amino acid, but some of them vary from, $100 like per milligram to thousands of dollars per milligram. And it's really not feasible to, only get a 10% improvement in potency if you're spending 10x on the acquiring that building block. So this is really only useful when you have a big library of chemical building blocks available to you, but it's something that, is not that hard to actually have as part of your process, like at least an understanding of it. In the biologics development space, there's been tons of nice retrospective studies about what biophysical filters led to successful clinical stage antibodies. This one from Jane et al., that's like an Adimab study, did a nice job of looking at biophysical properties, some of which you can compute computationally. I think there are some computational metrics in this process, but I think these are also experimentally measured properties, where sometimes you can see quite a range in what's allowed in order to sort of see if your antibody pushes the envelope or is kind of within the pack of standard, melting temperatures and things like that. So yeah, like that's always like an interesting thread, is like, can you predict as early on things that will help you helpful downstream? A simple thing that's frequently done is looking at aggregation prone regions. It's a little bit like that solubility surface examination we looked at as a previous habit. This is just an example recently in the MAPS journal of, reducing an aggregation prone region to, they demonstratedly improved CMC properties. They actually like kind of followed through 2 antibodies and looked at how much could be produced based on, these little plots don't tell you much, but they demonstratively improved those overall manufacturing properties downstream. Okay, we're getting through. Maybe a bit too fast. So this is like just a good one to put some context into this whole thing, is understand, then be understood. So you genuinely want to understand others, in this case, others like your drug target of interest. Maybe this compel them to reciprocate is kind of hard with the metaphor, but to create an atmosphere of caring. You want to be kind of like intimately in tune with your drug target of interest. And for that, it's kind of simple. You need to get an understanding of them. So successful protein therapeutics emerge from, before we get into that, just like a lot of foundations of knowledge of chemical probes, whether those be small molecules published in academia or larger biologics that are already designed for that target. Looking through vendors of antibodies, there's an antibody for almost everything these days. Sometimes the epitope might not match yours, but sometimes there's the benefit of having crystal structures available of small molecules bound to your target or larger biologics, which give lots of insight, even though you might not be able to have such a complicated binding mode as a small molecule, as a protein therapeutic. I mean, unless you dip into the non-natural amino acid catalog, that's just a great benefit to you. Great therapeutics kind of would engage in this characterization of the target, either through structural analysis or even biochemical sort of experiments to inform the drug design process. So it's not that every project needs this type of approach. Workflows in pharma for developing biologics can omit some of this structural detail, but structure-based design kind of thrives in this space. Like you can see some of these techniques on the side, which of course always help in stages of drug discovery with large number of quantities. They're high throughput screening methods. And you can go down to fragments and hit delete optimization with smaller quantities, smaller libraries. Let's give you some examples of those. So when I'm talking about tool compounds, like there's 2 examples, I guess, of starting hit-based libraries. So these might not be, these might form the foundation for a very successful therapeutic. That is screening structurally diverse libraries against your target. There's now, lots of great examples of both of display methods like phage display or, intracellular display of antibodies or other antibody formats. Well, here is an example specifically of interest to protein cure about cyclic peptides. And there are companies like Bicycle Therapeutics, which have used these methods routinely to screen targets. And there are even examples recently like this one, the development of a cyclic peptide binder to KRAS G12D from the UCSF team and the Suga lab at University of Tokyo. And that's really, although this peptide specifically didn't have some of the properties necessary for hitting this target in cellular assays, it was really just a very valuable tool for in vitro binding assays. So Yeah, that's starting points. And, doesn't always have to be these peptide style of things. I wanted to give this example to the right of a mini protein library of very specifically like a DARPins library that is, there's numerous non-antibody scaffolds. This one's a really nice one that's been used or studied extensively by the Rosetta protein design community as well. Um, and so that is another tool for starting design, although the DARPins might need further development or further, refinement. It's really, again, yeah, that starting position that's helping this. So talk about characterization now of the proteins or the protein-protein interaction. It's not, I guess, very routine that you have a method so intense as a deep mutational scan of the uh, peptide, or in this case, a peptide and receptor interaction, but it can be done through some experimental procedures like this one by Joseph Rogers, and that's PUMA with MCL1. So, that's a very rich data set right there for, it's even goes beyond the natural amino acids to non-proteogenic, non-canonical amino acids. It kind of gives you this kind of exhaustive landscape to explore, and your protein designs could be heavily informed by that. Even if it's not a single alpha helix that is the most stable thing, it's like you could use this to build a helical bundle or some other strategy of that kind. And there's about a zillion different studies on actual protein structures and dynamics of targets, the molecular structure and function of GPCR. Activation is shown here, at least for class B GPCRs that bind, in this case again, helical peptides. You can see all sorts of extracellular loops flopping around, some differences within these peptides within this family. It's only thanks to many cryo-EM structures and many labs around the world that we have this type of real insight into what kind of flexibility there is in the binding site, as well as the downstream result of that, of those different binding modes. In GPCRs, there's basically everything you could think of when it comes to complexity. There's allosteric interactions throughout the protein, communication from extracellular to intracellular. There's ion binding positions and structural water molecules all over the place. And there's bias signaling. Basically, you can have protein ligands bind at the top of the protein and induce different signaling pathways inside the cell. So that's just one target class. So I'm not saying that every protein therapeutic needs to know the whole story, but it certainly makes the job a little bit easier to think about at least active, inactive, and intermediate states of your protein if it has a well-defined kind of dynamic life cycle. Great. So next, think about synergizing. So that's combining the strengths of people. In this case, people may be other proteins through teamwork. So to achieve goals that you could have never done alone. It's sometimes the case where a single protein alone can't get the job done. And antibody, the biologics community has really knocked that one out the park by coming up with about every single different antibody format that you could think of, like taking and stitching together every single part of IgGs in different ways in order to get different types of interactions, whether those be maybe just uh, like a homo oligomerization, like in this case, maybe the binding epitopes is the same on either ends of the antibody, or maybe they're different. It's a bispecific that can bind to two different epitopes, and that could have a very important functional consequence depending on the particular disease indication you're going after. And there's even just assembling these, in this case, antibodies together in these really large, formats to get this avidity effect. Why have one binding site or one binding interface when you can have 10? As long as you can stably display them, you might as well go for more. You can potentially pick up more interactions, and just the net result is you get overall better binding affinities when in your experimental measurements. So, binding doesn't just stop at binding though. Binding, or maybe even this bispecific interaction, which allows you to engage two targets in two different ways. You could also recognize that, attaching yourself to or conjugating yourself to small molecules or other proteins could allow you to have new functions. So as a protein therapeutic, don't set the bar too high for yourself. You could allow the dirty work to be done by a small molecule to get the job done. A couple examples of that. Multivalency for increased avidity. Here's an example of the SCFE. That's like just the binding end attached together, and it's tetravalently linked in this picture. Specifically, this is Humira, one of the most successful antibodies on the biologics market. And you can see that at least this response is, as good as Humira itself in this tetravalent format. So there's probably better examples of this too, but many platforms have been developed to display antibodies in different ways. Tetravalent, there's octovalent, there's different ways of linking them to stable proteins that flow through the body. And that whole idea of using bispecifics is a hot topic these days. Multi-specific binders. It exists, or it has existed in the peptide world for a long time. Dual agonists to GPCRs. But certainly it became especially exciting in the field of immuno-oncology, the development of multi-specific binders for dual target engagement. This example by Amgen of this Blincyto engages a cancer cell surface protein, CD19, and it also interacts with a T cell receptor. So this type of bridging the gap, bridging these two interactions, allows for the T cell to kind of handle these cancer cells. It kind of redirects your immune system towards these proteins. These cells. And now getting to that point about antibodies not doing, or biologics not doing everything by themselves, there's a point in which you may feel that your protein serves the purpose as a delivery vehicle or a binder, but it actually doesn't do the get the complete job done. Like antibody drug conjugates are highly effective for delivering cytotoxic payloads, like this particular small molecule shown at the top. It would basically enter the cell and in this case bind tubulin and result in devastation, mass devastation to cancer cells. So that is obviously something that you can do in many different ways. Protein drug conjugates and peptide drug conjugates also exist to a certain extent. One place in which you see a conjugation to peptides is through radiopharmaceuticals, like this example on the right, where we have a cyclic peptide that binds somatostatin. And the example here is this, radioisotope, which is allows you to image tumors highly effectively. And the benefit of really building radiopharmaceuticals with proteins is that proteins, can be degraded. You don't have to be the most stable protein in the world to serve your purpose as allowing imaging of cancer cells. So that is a place in which, proteins can definitely recognize their limitations. They don't always have to be the best binders. They don't always have to be the most stable with respect to, some competing platforms. But, you got to know your place. Okay, so we're coming to the end now. So this is just like a general overarching principle that is really important for drug discovery in general. Balancing, renewing your resources, energy, and health to create sustainable, long-term, and effective lifestyles. So that's pretty vague, but in general, it means that you need to iteratively develop. Classic concept in drug discovery, I think is a good one to end on, is that ultimately protein therapeutics, like all of the proteins, or like all of their therapeutics, succeed through iterative development, computationally and experimentally. That's the faster you can sort of catalyze the development through fast design, make, test, analyze cycles, the better you are. And whether that's assisted through human or maybe even machine-guided active learning for understanding what changes you should make to your structure in order to have that activity response, that is just great. And it requires often lots of assay development, and to do things in a higher throughput or, big scale. The promise of computational methods and design is that there's a possibility that you can not necessarily have to do such large high throughput methods. You could just synthesize a small batch of proteins and hopefully enrich on your hit rate. But inevitably, in the big scheme of things, iteration is important. We're not quite there yet in the tools and workflows. These strategies work for some proteins. They don't maybe work for all proteins. But it's generally the best you can do if you can seek an experimental platform for testing that resembles kind of in vivo, or ideally like a clinical scenario. But you obviously can't simulate a patient. But you can try to account for some of the complexity and problems like immunogenicity. You really do know, based on, sometimes you do know what kind of immune, I guess, MHC or HLA alleles are in the patients, such that you can kind of know or test across enough of these that you might get an indication of what your drug might be like in a real patient population, just like an anecdote there. So I don't really have tons of examples of this, again, because platforms are often a little bit behind closed doors, but Protein Cure works to improve our design, make, test cycles. But ultimately, all the strategies come together with this one. You basically try a lot of these habits and work towards the future. So to be best in class, I've described to you some habits for success. These ones that are based on independence, doing things yourself, being proactive. That means getting yourself out there in the body or to your target. There's the one about beginning with the end in mind. Like, that's when you have the opportunity, always think about what epitope you want to bind to, what type of target, what kind of shape of a protein you want to be. And putting first things first is obviously a big problem when multi-objective design is such like a headache-inducing process. You certainly have to prioritize, because otherwise you'll be moving forward and then moving backward at the same time if you're not careful. So interdependence is also important habit for success. You of course want to think win-win. That is, think about the bigger picture. Often these business objectives like scale up or manufacturability, things that you might want to consider. Seeking to understand before being understood. Remember, that was about knowing your protein target through ideally some sort of deeper characterization that you would normally have. Gives you an edge, and it helps you become more in tune with your target. That whole idea of synergizing, that's working together with other proteins to become, better binders or conjugate with things that do functions that you wouldn't normally be able to do. And ending on really continuous improvement. That is iteratively growing as a therapeutic. It's that's the way it has been done for a long time, and the way it'll probably always continue to be. It's just how fast we can do it, which ends up being sometimes a bottleneck for progress. So after you win the Best in Class award, you can achieve your aims. That's kind of wrapping up to the end. I just could end off by saying that Protein Cure is based in Canada, and we develop and apply methods like protein design methods, machine learning, and we have sort of an experimental pipeline as well. We do that with our partners in pharma R&D as well as internally. We're always looking to expand our team. So thanks to the team behind me, as well as our investors and our collaboration partners. But that is it. And if you ever get a chance, you can read my the books. It'll soon be on shelves. Yeah, soon to be a bestseller, over 15 million sold. That's just a projection. It might not have a huge market, but as the original has habits of highly effective people, but who knows? So yeah, with that, I will take questions anywhere throughout. Happy to.
Awesome. Thanks, Chris, for a really nice broad overview, providing some really good perspective on the field, really. yeah, let's open it up for questions for Chris.
Maybe I can start with one that's more the big. So I think if you leave antibodies for 1 second on the side and anything that's engineered of antibodies, right, most of those protein therapeutics has remained somewhat one-off successes. So that doesn't speak, it's really this individualized, right? The one thing that I find surprising, for example, is the stable peptide that's compelling on the first, right? And first impression, it's compelling as a concept. It really hasn't gone beyond the one aileron so successful. I wouldn't call it a success. And there could be two reasons. There could be it's just a very bad target choice, and I would probably be in the camp of supporting that notion, and maybe that's prevented anything to follow. But yeah, but kind of general thoughts on how much, and I think delivery certainly is a big piece of the whole equation when it comes to intracellular targets. But it seems like we really haven't figured out how to generalize some of the concepts. Much of the cyclic peptide stuff has the peptidreams, all those once promising approaches have kind of gone to shelves to some degree. I don't know. Yeah, just general thoughts.
No, that's all really good point. Like it has been a while since some of those technologies have been developed that have let peptide therapeutics be screened on a larger scale, which should have resulted in, lots of exciting things that we haven't seemed to come to fruition in a lot of things entering clinical trials. A lot of the peptides we do see that, beyond this aileron peptide, we're kind of in the space, so we follow a lot of these things. There are certainly a lot of success in peptides that are close to hormones, naturally occurring peptides. It's really the de novo side of things, which gets you wondering, like, things just from scratch are few and far between. So we haven't really seen the applications reach that stage, and it's a matter of time. I think that we are like getting more, I guess I'm a bit biased, but we're getting better computational models and more data than ever as part of being generated on these things, which, yeah, just takes the right team to put it all together. I think that intrinsically, there's no reason why you can't have a cyclic peptide achieve the things that we've seen small molecules done so well, but sometimes there's just a bias towards those small molecules because of the workflows and staff and the procedure is so set in stone. So there's kind of like a structural element to it also. Same with biologics. It's kind of either of those camps. You kind of have a workflow in place, but as soon as things start getting a little bit beyond rule of 5 and a little bit uncomfortable, it just becomes too risky. It's like, why take the risk on some things? And an example of why aileron is like such a difficult problem is that it was kind of like an undruggable target where nutlins and other various small molecules failed. So it tends to be that peptides and proteins get stuck with the hardest, most well-established targets, but the hardest ones where there's no other option available. So they kind of get roped into this tough situation.
I think and then I'm just, I'm a fan of proteins in general, and part of why I'm critically questioning the spaces because I'm trying to figure out what really we have to do to move this to the next step. I think if you look at the aileron peptide, for example, it doesn't look like a peptide anymore from a chemical structure. I followed very closely back then, still being part of a large pharma organization, some of the trying to progress hits from cyclic peptide screens to actual cell-permeable, bioavailable molecules. Again, they don't look anywhere near to what they initially looked like. All of the advantages of designability and selection kind of go away in this process. And I think that's, to me, one of the biggest challenges that we haven't really solved yet. How do we put some of this effort up front so we can either design more tractable or we can select more tractable?
Yeah, that's a big problem. Yeah, and I think that, yeah, there's no easy answer. It's going to be just the grind, pushing things forward on the basic science level, but also on the application side.
Yeah, yeah. I didn't want to start as the devil's advocate here.
No, that's really good.
I have a related question that fits to that. So one aspect, I'm at Biogen, I would say, is immunogenicity. So with all these exotic things, stapled peptides and new folds and stuff like that. In one of the first meetings, somebody would ask, what about immunogenicity, right? And so is that something, Chris, that you see in your work with larger companies that's a concern? I mean, you pointed to, okay, you tend to use these for tried and true targets, and so other stuff has failed, so people are maybe more risk taking, have a more risk forward stance on that. But what are you seeing there? Is immunogen? It's hard to test. You can't stick it if you stick it in a mouse. The mouse is not immunogenic, or it is immunogenic. It doesn't tell you anything about the primate later, and so forth. And really, only the human trial will tell you. So that's sort of the big headache with immunogenicity.
Yeah.
So what are you seeing from your partners there?
Yeah, it's difficult. Like on the immunogenicity side, it's sort of more in the natural proteins world. I would say, like, I guess we were connecting it a little bit to the previous point about the non-canonical amino acids and staples, which have their own sort of problems associated with them. So I think that's not something that's easy to predict. I guess thinking from the machine learning perspective, there's just vast amounts of MHC binding data at the IEDB. There's now, lots of sequences, like billions of sequences of antibodies in the immune repertoire, but they still don't give you a full picture. Like they're data sets that are some of the best and largest data sets available for peptide interactions, peptide fragments to MHC for MHC display. But again, linear peptide motifs and T-cell interactions is basically like only one side of the battle. Like you get immunogenicity based on aggregation, based on all these meta properties. So yeah, it's a big challenge that pharma partners don't really have a lot of solutions to. Like I did a survey sort of what commercial offerings are available for companies working on biologics, and they're not that advanced. Like the dehumanization papers that have been written a little bit in the protein design world, some I think like is it Griswold and Kellogg did like some nice work on this. Actually, in this chat, I believe we have the MHC epitope energy author Graham here. But anyway, yeah, like the methods are not that robust for, I would say, it's an unsolved problem.
It's still unpublished, so I don't know if I count as the author yet, but.
Yeah, well, I mean, yeah, it's contributed at least to the documentation, so published for all intents and purposes.
I mean, immunogenicity is going to be a big issue. I'm also curious to see if immunogenicity is going to haunt us with covalent molecules becoming more popular. And once you move out of the space where you are right now, which is oncology with oncogene mutations, right? Suddenly you will create neoantigens. And I wonder, for example, the reason we have so many allergic or allergies against beta-lactam antibiotics is because they're highly reactive. Right? So, and it's just not a problem right now. All the molecules we're putting into the clinic, no one is going to worry about a KRAS G12C neoantigen. That might even be beneficial. But once you move this into general medicine, that's going to be, I think it's going to be a topic.
And their peptide therapeutic symposium this year was kind of like a little bit, some good examples of the translational side of those companies in biotech working on, really these downstream applications of peptides. Like you obviously don't hear about the things that don't work though, as is the problem always. It's hard even to assess from the outside why things like that are well studied, like the aileron peptide, which seem to have lots of promise, didn't achieve on some of like the endpoints that they were working on, which is it's sort of pivoted now, and there are now like successor developments through companies like Fog Pharma Greg Verdeen. So they've really built a platform that helps to get both to the bottom of the permeability problem through high throughput, basically log D measurements and tons of SAR on things, understanding why things go through cells and some don't, along with just a display screening approach. So I think they're generating lots of data. That's one thing that I hope we're doing better than we ever were before. That's why I'm optimistic. And as long as you're kind of keeping up with the machine learning side of things, you're gaining a better understanding of sequence-based data through natural language processing and also structural data through the really nice work on, like structure-based ML. So, I mean, I tried to balance a little bit about the antibody side of things, which is kind of a big topic unto itself, and the peptide world. The mini protein world is an interesting area where I guess, Chris, you're kind of a little bit more focused on, where I hope we'll see more of these mini protein scaffolds functionalized for purposes as binders.
We do.
Well, they, I guess, haven't been put through the gauntlet of these immunogenicity checks either.
Yeah, just mice so far, which doesn't count.
Well, that seems like that's at least better than that's good.
How
start.
about bioavailability and permeability for those?
You say oral bioavailability? Well, I mean, that's the dream. Yeah, we don't know that yet. One thing I'm optimistic about is there's a pesticide company called Vesteron, and they use a mini protein they isolated from spider venom as a pesticide, and they use a food grade yeast to produce it. And if they can make this thing cheap enough to spray it on fields, then you can make a lot of it. And so the oral bioavailability it really becomes a numbers game, right? Like if 99% of it gets degraded, well, if I could add 100 times excess of what I need, and it's just protein, so there's no toxic byproducts from the body breaking it down, you can give someone a protein shake.
Yeah.
That seems like a Ginkgo Bioworks type of thing. They're building these kind of, factory applications, and they partner with some of these ag tech or agriculture companies.
Or like the drinking 100 liters of red wine a day.
For, was it resveratrol, right? That's the it's like you'll live forever until you die of liver cirrhosis.
Yep. I mean, if you put your mini protein into beer through the yeast, then
Well, I mean, I guess we do a survey of like the approvals of peptides and protein therapeutics, and things just generally don't move very fast to see these examples, but there's certainly lots of examples of the oral bioavailability improvements on the the type 2 diabetes side of things with Novo Nordisk.
I mean, to kind of counter my skeptical first comments, my general thoughts are that there's tremendous pressure and tremendous incentive to ultimately solve these problems, and so we'll find the solution. I don't know what it's going to be, but I think we'll solve it ultimately.
Just speaking of go for it.
Oh, okay. Actually, it's just like a really half and probably poorly thought out idea, but I mean, have there been any drugs that work off of functionality that sort of, in terms of, you're saying sort of getting through oral bioavailability, to what extent has there been work done on hijacking IgA transport?
Mm. I am not too familiar about that space. I know this conjugation to IGFCs works well, but I don't really know much about that. GG transport. Sometimes you just want to be generally the right size of an antibody or size of your therapeutic such that you don't get cleared by the kidneys. So there's like an interesting just size range where maybe you can get transported around the body better just by being a certain Daltons. That's just like a general catch-all thing. Okay, well, I don't want to prolong this longer than necessary, but I mean, again, still happy to talk about any things in this space. Anybody could reach out to me if they have any interest in proteins or protein therapeutics and how that intersects with computational methods.
Cool. Yeah, let's thank Chris one more time for awesome seminar.